Hi, my name is Morgan Robinson and this is my video for Exam 5. A protein that is 4 amino acids long is formed from this DNA sequence in vivo. We need to determine which sequence is the template strand and which is the coding strand in order to transcribe the DNA into RNA. A way to do this is to flip the bottom strand so that it reads from a 5 to 3 direction. In doing this, we can see that there is a start codon in both strands, which is ATG. Now that we know where the start codon is, we can determine which strand is which. The stop codon will be 4 amino acid sequences down from the start codon, which is the coding strand. In the top strand, TGC is the fourth one down, but this is not a stop codon, so this cannot be the coding strand. In the bottom strand, the fourth one down is TAA, which is a stop codon, as the T will change to a U, and UAA is a stop codon, so we can determine that our bottom strand is the coding strand. Now that we have identified our coding strand, we can use it to transcribe the DNA sequence into mRNA, and to do this we just switch the T's to U's. Using the mRNA sequence, we can now translate this into an amino acid sequence. With this strand, we would start with AUG, which codes for methionine. Next would be GUC, which codes for valine, then AGC, which codes for serine, AAG, which codes for lysine, and finally we would have UAA, which is our stop codon. That tells us to stop translating. Now that we have completed translation and transcription, we can identify what would happen if there were different single nucleic acid point mutations in the coding strand of DNA, and how it would affect the translated amino acid sequence. First, we will start with the missense mutation, which is where the change of a single nucleotide results in the substitution of a different amino acid sequence. To illustrate this, we see a transversion that occurs where the G is switched to a C, changing the amino acid sequence where ACC codes for threonine instead of serine. Next, we will illustrate a nonsense mutation, which is where the change in a single nucleotide results in the appearance of a stop codon instead of an amino acid. This results in a shorter, less functional protein. We can see the example here where a transversion occurs and the A is changed into a T, which eventually becomes a U after transcription. Our amino acid sequence then changes to UAG, which codes for a stop codon earlier than originally planned. Next, we will illustrate a silent mutation, which is where the change in a single nucleotide results in no change of the amino acid. We show an example here where a transition occurs and a C is changed to a G, which changes our amino acid sequence to GUG. However, the strand is unaffected because GUG still codes for valine. Finally, we will illustrate a read-through mutation, where there is a change in a stop codon, which converts a stop codon to one that codes for an amino acid. This results in the extension of the polypeptide chain. As an example, we will do a transversion of an A to a T, which eventually becomes a U after transcription. This results in a change in the amino acid sequence, switching it from a stop codon to the amino acid, leucine.